For the animal animal cell culture is a current method, protocols, and technology that will give rise to a possibility to grow, proliferate, have a stock of animal cells in vitro, so in a lab. To start, I want to explain the animal cell morphology in vitro, so observed in a microscope, okay, under a microscope. Animal cell morphology technique is a simple and direct identification of the cultured cells, and it can be done by observing their morphological characteristics under a microscope. However, the morphology has to be viewed with caution since it is a large dependent on the culture environment. For instance, the epithelial cell growing at the center of the culture are regular polygonal with clearly defined edges مثل ما نكون عم تشوفوا هون the epithelial cells under a microscope, optic microscope. With the cells, they are grown in the periphery على الطرف are irregular and distanted. In a tissue culture in the lab, we use two terms. The term fibroblastics and the term epithelial are commonly used to describe the appearance of the cell rather than their origin. ما إلهم علاقة من وين cells جايين، ممكن يكون endothelial origin، ممكن يكون mesoderm origin أو source of the cells، بس we describe their morphology using fibroblastic-like cells or epithelial-like cells. So the fibroblastic-like cells, for this one, the length is usually more than the twice of their width. يعني هي بتكون طويلة cells like a fibroblast. The fibroblastic cells are bipolar or multipolar مثل ما عم نشوف هون in nature. For the epithelial cells, these cells are polygonal in nature with regular dimensions and usually grow in monolayer. The terms fibroblastoids, so fibroblast-like cells, and epithelioids, يعني epithelial-like cells, are in use for the cells that do not possess specific characteristics to identify them. Okay, so نحن بس نفوت على اللاب فينا نشوف we can observe our cells under microscope ونقول إنه they are fibroblastic like cells or epithelial like cells بس ما إله علاقة بالcharacteristics بالfeatures biological one of these cells. The animal tissue culture can be divided into organ culture, tissue culture, and cell culture. Okay, donc three types of animal tissue culture أكيد in vitro عم نحكي. هلا entire embryos, organs, or tissues are excised from the body either by vivisection or shortly after brain death ويكونوا هن source of organ culture. In such cases, the normal physiological functions or organs or tissues are maintained أكيد time dependent. The cell remain fully differentiated and scaled up is not recommended in organ culture. Our aim is to put this organ or tissues يعني organ viable على وقت محدد for future uses. وأكيد الجروث بيكون كتير slow لأنه ال differentiated cells are in a extracellular matrix and they have a rate of proliferation كتير واطي like a tissue in vitro. The second type is the tissue culture. This type of tissue culture we can obtain when fragments of excised tissue are grown in a defined medium. Most of the normal functions of the cultured tissues may be maintained even though the original organization of the tissue is lost. And this type of tissue, of culture, tissue culture, يعني, is better for scale up but not ideal because we have a fragment of tissues. The last or the third type of animal tissue culture is the cell culture, when the excised tissues or tissue outgrowth are dispersed, and a dispersion, mostly enzymatically, into a cell suspension. So we have single cells, which may then be cultured as a monolayer or suspension culture. The cell cultures can be maintained for very long time as a particular type of the cell line and are very switchable for scale up and studies. So we can pass the cultures and we can have in one petri dish, we can have several one. So we can scale up easily. Some features of animal cells grown in vitro. You know that animal cells, unlike plant and microbial cell cultures, show limited growth even if the best of nutritive and physical conditions are maintained. The behavior of animal cells in culture depends on the source organs or tissue. Why? Because we have a type of tissues that maintain a high proliferative rate in vitro and in vivo, and we have other type of tissue that can divide one or twice لأنه it's a specialized cell type like neuron. Neuron tissue بتعرفوا إنه neuron cell is a very specialized, highly specialized cell, so division تبعتها is very 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 low والregeneration كمان. And I give you a high proliferation rate example, the epithelium, intestinal epithelium مثلا, it can divide several times and with a proliferation rate very high. One. Now I will explain the very important graph of proliferation and growth of a animal cell culture, animal cell in culture, in vitro culture. Typical growth curve, like you see it here, curve for culture cells display a sigmoid patterns of proliferation. I will explain. The growth phases associated with normal cells are defined as lag, log, stationary, yani plateau, and death. Firstly, cells are in a lag phase. So when we put cells in a petri dish with the culture medium, the supplementary one, the complete one, cells will be in a lag phase, usually no more than 1 to 2 days length, أكيد منه أكتر, during this phase there is a little or no increase in cell number. عم نشوفها واتي تحت. During this time, cells are conditioning the medium. عم يتعودوا على النيو أو الفريش medium اللي نحطه فيه. Undergoing internal cytoskeletal and enzyme change and adjusting the new medium to discover which molecules are in this medium, which types of growth factors or cytokines, etc. in this medium, and it will condition this medium. So in the lag phase, between 1 to 2 days, cell will condition the medium. Second phase is the log phase. In this phase, cells undergo a period of a rapid division, so proliferation rate عالي كتير, so-called log phase growth. Note that each cell line will show different cell proliferation kinetics during the log phase. حسب التايب اوف سيلز. اذا هي بالسورس تبعتها in vivo she has a high rate of division of proliferation، واذا اعطيتها انا الميديوم القريب عليها اكثر شيء she will conserve this rate of division. like الاكزامبل اللي قبل شوي يعطيناها عن النيورون وعلى الابيثيليال سيل. So after this proliferation rate cells will increase in numbers يعني عن proliferation phase they will increase in number and approach a confluency and form contacts with another cells. يعني بدهم يكونوا كتير قراب على بعض وعبوا كل الباتري ديش، صار في عنا شيء بنسميه confluency. This confluence the rate of division slow and they are most susceptible to injuries. This is the stationary phase, the plateau phase, when cells are confluent وأقرب للستريس أكتر ما تكون أقرب لللوج phase. So بيصير في عنا contacts between cells وهون في عنا a high risk of inhibitory contact ويقبعوا السيلز ويموتوا، so cell death or not. So إذا خليناهم عطول عنا a rate of proliferation inhibition contact cell will undergo apoptosis وبدها تموت. If not we can proceed to passage this cells ونرجع ننقلهم and divide them to a new petri dish حتى نرجع على اللاج phase log phase وكمان etc. So في عنا عطول هالاهتمام بالسيلز every day أو every 2 days. Here I spoke على inhibitory contact. So شو هو؟ We have two ways to the in vitro growth of cells. Cells may grow as adherent, monolayer or in suspension. في عنا two protocols to each type of these cell growth. For the adherent cells, adherent اسمها مع adherent يعني عم تعمل adhesion on a surface, on physical surface. So في عنا anchorage dependent and attachment to a substrate. Substrate العادي الجنرال one هو ال surface of a petri dish of a flask culture for cells. So cells grow and spread in a monolayer when they adherent. Attach to the surface, fill the surface of the container or the flask or the petri dish. Once the cells spread the entire surface and come in contact with the wall of the plate or the container, the cell stop further growth. بده يصير في عندها مثل risk، بده يصير في عندها alert. The phenomenon is called contact inhibition. Most cells, with the exception of mature hematopoietic cells, are transformed cells grow in this way. لأنه transformed cells يعني cancer cells will grow in a behavior كتير different of this one, in a spheroid behavior ونرجع نشوفه بمطرح تانية. And the hematopoietic stem cells which are the blood cells, they are not adherent, they grow in suspension. So when the surface are filled with the cells لأنه خلص بطل في محل, the cells will have contact inhibition and they will be detached from the surface container, and will undergo a apoptosis pathway and cell death. If not, we can do an intervention here, and we can use enzymes, proteolytic enzymes, to break the bond between cells here, and we can retrieve these cells seeding them into a new petri dishes لأنه هون نستعمل أكتر، يعني صار في عنا عدد كتير كبير number of cells بنقبعهم using proteolytic enzymes, and we can like proteolytic enzymes like collagenase, papain, dispase, trypsin, pronase, etc. We can use these enzymes قبل ما هي السيل تعمل contact inhibition. ناخد السيلز and we can divide them into a new Petri dishes to increase more their numbers. So this step here is called the confluence, when